Topic: Meiosis. Aims of lesson. At the end of this lesson you should be able to know the following. Structure of a chromosome and associated terminology. Process of meiosis. Importance of meiosis. Introduction. Revise mitosis and cell structure with emphasis on parts of the nucleus, the centrosome and the cytoplasm. Observe diagrams/micrographs of cells in selected stages of meiotic division. Concepts and skills. Below are definitions of some of the terms that you will study under this section. Use the glossary in your textbook to define these new terms/words. Note, at least 2 marks are awarded if you can define a term correctly in a test or examination. Haploid cell, a cell where there is only one set of chromosomes. Diploid cell, a cell where there is a double set of chromosomes. Gametes, sex cells e.g. ova and sperm which have a single set of chromosomes. Somatic cells, body cells which have a diploid number of chromosomes. Gonosomes, the sex chromosomes in an organism. Autosomes, chromosomes that are not responsible for sex determination. Homologous chromosomes, a pair of chromosomes that is inherited from each parent, that have the same genes at the same locus. Cytokinesis, division of the cytoplasm during cell division. Study the following information on chromosomes, mitosis and meiosis. Structure of chromosomes. Chromosomes consist of DNA, which makes up genes, and protein. The number of chromosomes in a cell is a characteristic of an organism, e.g. humans have 46 chromosomes. Diploid cells have 2 sets of chromosomes, where each chromosome has a homologous partner. Haploid cells only have 1 set of chromosomes. Chromosomes in haploid cells have no homologous partners. Before meiosis begins, during interphase, DNA replication takes place. Chromosomes which are single threads become double, 2 chromatids joined by a centromere, as a result of DNA replication. Structure of chromosomes. Homologous chromosomes. Unreplicated chromosomes. Revision of the process of mitosis. Meiosis. Meiosis is a type of cell division whereby a diploid cell, somatic cell, undergoes 2 cell divisions, and divides to form 4 haploid cells, gamete or sex cells. Meiosis takes place in the testes and ovaries of animals and in the anthers and ovules of plants. Note, meiosis is a continuous process which is divided into the first meiotic division, meiosis I, and the second meiotic division, meiosis II. The events of meiosis are divided into different phases for convenience. Events of interphase. DNA replication takes place. Chromosomes, which are single threads, become double. Each chromosome will now consist of 2 chromatids joined by a centromere. DNA replication helps to double the genetic material so that it can be shared by the new cells arising from cell division. First meiotic division, meiosis I. Prophase I. First meiotic division, meiosis I. Prophase I. During prophase I, the nuclear membrane and nucleolus start to disappear. Spindle fibers develop between the centrioles to form the spindle. Chromatin network condenses into individual chromosomes. Pairs of homologous chromosomes lie next to each other forming a bivalent. Adjacent chromatids of homologous chromosomes overlap and touch each other at a point called the chiasma, plural, chiasmata, in a process called crossing over. Chromatid segments break off and are exchanged, resulting in the exchange of genetic material. This process is called crossing over and it brings about variation. Metaphase I. During metaphase I, the homologous chromosome pairs are arranged at the equator of the cell in a random way with the chromosome attached to the spindle fiber. Which chromosome lies on which side of the equator is totally up to chance. This is called random arrangement and brings about further variation. Anaphase I. During any phase the chromosome pairs separate and chromosomes move to opposite poles, one to each pole. Telophase I. During telophase I the chromosomes reach the poles of the cell. Each pole has half the number of chromosomes present in the original cell. The cell membrane constricts and divides the cytoplasm in half to form two cells. Second meiotic division, meiosis II. Prophase II. During prophase II the chromosomes are not in pairs. Nucleolus and nuclear membrane disappears. A spindle forms. Metaphase II. During metaphase II the individual chromosomes line up at the equator of each cell, with the centromeres attached to the spindle fibers. Anaphase II. During anaphase II the spindle fibers contract. The centromere of each chromosome splits into two. Two chromatids of each chromosome separate and move to opposite poles. Telophase II. A new nuclear membrane forms around the unreplicated chromosomes at each pole. Cytokinesis splits the cells into two new cells. 4 cells, each with a haploid number of chromosomes, have been formed. The importance of meiosis. Production of haploid gametes. The having effect of meiosis, from the diploid to the haploid number, overcomes the doubling effect of fertilization, thus maintaining a constant chromosome number from one generation to the next. Meiosis introduces genetic variation through crossing over and the random arrangement of chromosomes at the equator. Common errors made by learners in examinations. Unable to answer and interpret questions based on chromosomes and meiosis. Unable to correctly describe the events during each phase of meiosis. Unable to interpret and answer questions on meiosis based on given diagrams. Thanks for watching. Please do not forget to like, share and most important subscribe to the channel.